Okay, so today is our 15th lecture. So we finished the single cells analysis part, and now we are moving beyond transcriptomics into proteomics, it's study of proteins. And we need a new tool. Like we no longer can do sequencing technology. Now we need to use mass spectrometry. So today we are going to cover these two topics, like the study of protein and the mass spectrometry instrument. Okay, so that's our agenda today. And one extra content will be about how to use mass spectrometry to study protein structure. So not just expression level, but we can also analyze protein structure with this technology. So first, just want to make sure that we know that proteomics is not just about protein expression. It's a study of protein. So protein has function, it has post-translational modification that can affect how it operates in the cells. Right? It has 3D structure, so two proteins can bind together. Right? Those are also important to understand the protein. But most of the time, when we talk about proteomics, usually it's about measuring the expression level of proteins. So it's similar to transcriptomics, but at protein level. So one of the key questions is why don't we do proteomics all the time? If we want to understand biology, function occur on the protein, not on RNA. So the most direct evidence would be the expression of proteins, not RNA. There are many limitations. First, for DNA and RNA, we can amplify with PCR. But for proteins, the amount of material that we got from the patient or from the tissues, that's everything we have. We cannot amplify further. We have to maybe culture more cells or get more blood from the patient or biopsy more tissues from the patient. And those are costly. Those are invasive procedures. The second difficulty is more on the wet lab side. For DNA and RNA, all the molecules are similar. It's just nucleotide. There is no complicated structure or chemical properties. But for proteins, we know that amino acids have different side chains. Some have a positive charge, some are negative charge, some have the aromatic ring. So they are heterogeneous. And that creates a problem when you want to extract protein. Some protocol might not be appropriate for hydrophobic protein. Some protocol might be appropriate for polar protein, for example. And some proteins are also integrated into cellular structure. So it's not like you get the extract of the cytoplasm and you get to see all protein. Some proteins are integrated into the cell membrane. Some are imported into other organelles. So it's not easy to extract every protein that you are interested in. And the most important factor that why don't we do proteomics all the time is that we don't have a direct technology to read out the individual amino acid in the sequence. Unlike DNA, where if there is an incorporation of a nucleotide, we understand exactly what's the nucleotide on the template. But for proteins, we don't have a polymerization reaction. So we cannot read out amino acid one at a time. We have to do something else. So we have a problem that's similar to nanopore technology. Nanopore technology utilize the structure, change in structure of the DNA or RNA strand. So they don't have the readout for individual nucleotide. So they have to train deep learning model to process the signal. So the same challenge is also occurs for proteomics with mass spectrometry data. And also, to make the problem even more complex, for RNA and DNA, we have some isoform. One gene can give rise to multiple RNA isoforms based on the splicing. For proteins, as we will see, because we use mass spectrometry, as the name implies, we are going to analyze the mass of the protein. And all the post-translational modification can change the mass of the protein. So even if you have the same protein sequence, same amino acid sequence, if you have some change in PTM, the weight will change, the mass will change, and the data will look different on the mass spectrometry. So you have an even more diversity of protein molecules, not just from the sequence, but also from the modification. And from biology, we know that there are many modifications inside the cell. And many of these have function, like phosphorylation, can activate or deactivate all the kinases. So these PTM can dictate protein function. So if we don't know about them, like if we cannot detect them, then we don't have a complete understanding of the proteins in the sample. And just to give you an estimate, like people want to show how complex the data became. Like when you analyze a genome, you have about 20,000 genes for human. On average, these 20,000 genes can give rise to 100,000 RNA. So this is an isoform that came from splicing, from alternative splicing. But when it became proteins and you add modification, you add everything into it, you have 10 times more different proteins in the cells, up to 1 million possibilities. And each of these will give rise to different signals in the mass spectrometry. So the amount of complexity and the difficulty of data analysis also increase when we switch from RNA to protein. Okay, so up to this point, you might be curious about how we identify amino acid sequence from protein. Because we don't have the polymerization reaction, like what can we do? So let me give you the sketch here, and then we'll go over the entire process in the mass spectrometer afterward. So let's say you have a peptide. Peptide is a polymer of amino acid. Assume that these are the amino acids A, B, C, D, F, G, H. What we want to do inside the mass spectrometer is we want to generate the fragments or partial sequence, partial molecules from this peptide. So each peptide molecules, we want to fragment it into smaller pieces. And what we are going to see is we're going to see the prefix or the suffix of the peptide. So prefix means the amino acid in front of the peptide. And we are going to see A, amino acid A, like by itself. We're going to see a dipeptide with two amino acids, A and B, together. We're going to see a tri-amino acid A, B, C together as a molecules. So basically, we're going to fragment these peptides. There are multiple molecules of the same peptide. Some molecules will be fragmented between A and B, so that we produce A. Some molecules will be fragmented between B and C, so that we produce AB. So we are going to break the peptide bond that connect the amino acid together, so that we get to see all these fragments. And then, when we measure the mass, like we use the mass spectrometry, to measure the masses of these fragments, like the weight of these fragments. Let's say we get 10, 15, 22, like these are all imaginary numbers. So what can we deduce from this observation? Like all the things we see from mass spectrometry will be just a set of numbers. So what can we interpret in this like an idealized situation is the smallest mass is 10. And we know that the smallest mass is probably the first amino acid, because that's the smallest fragment that we can generate. So we say that the first amino acid, whatever it is the one that has the weight of 10. So we can look up which amino acid has the weight of 10. These are imaginary numbers. And then we know the second ion, the second mass that we found is 15. It's 5 more than 10. So we know that the second mass, the second fragment that we can see that's small, could be AB. So we know that A has a weight of 10 and AB has a weight of 15. So we can infer that the second amino acid B must have the weight of 5. And we can do a lookup table which amino acid has the weight of 5. And then we see the third ion, the third weight, being 22. So we assume that if this 22 corresponds to the weight of the third smallest fragment, which is ABC, then amino acid C has to have a weight of 7, the difference between 15 and 22. So we're going to perform this kind of a logical inference from mass spectrometry data to tell what's the first amino acid, what's the second, what's the third, by looking at these weights, looking at these masses. The problem is, when we look at real data, we are not going to be able to guarantee that we can generate all possible fragments. So there will be some missing numbers in this observation. And also, when we analyze a protein or a peptide, it's not a pure isolate. There's going to be maybe a different peptide with similar mass or some chemical contaminant or background signal that interfere with the measurement. So you're not just going to see like 10, 15, 22. You might see 12, you might see 8, you might see 17, which are not coming from this peptide. So the challenge in identifying peptides from mass spectrometry data is to make inference, like make deduction from this list of noisy measurements. Okay, so let's see what happens. Like how do we follow that kind of process, that logic process with the mass spectrometry. So if you haven't seen a mass spectrometry before, it's about as taller than a human. And inside, this is a purely physical instrument. So the peptide, once it enters the mass spectrometer, it's going to enter as a gas. So we start with, so we know that proteins are solid, right? And then we dilute them in solvent, in aqueous. And then when we inject them into the mass spectrometer, they will be turned into gas. And when they are turned into gas, this gas will be charged with some ion, some plus or minus charges. Inside the machine, they're going to try to measure the masses of these ions, each representing a protein, a peptide, or maybe a fragment of a peptide. And what they are going to measure, at first, is going to be the ratio between mass and charge. And we will see why it's not measuring mass directly, but mass overcharge. So the data that we're going to get from a mass spectrometry will be the mass spectrum. As the name implies, we are going to look at the spectrum of some masses. So all these numbers is telling us that in this protein or in this peptide or in this kind of mixture of protein, there are ions with a weight of 445.1218 units, and this is the amount of that ion. This is the abundance of that ion. And there are also ions with 705.8519 masses overcharged with a 2 charge on its molecules, and this is the abundance for that ion. So basically, a mass spectra is like a scale. It's going to measure every protein and peptide, every ions that exists inside the machine at that time. So it's just measuring mass and charge. There is no distinction whether some of these may be peptide, some of these may be other compound. It's going to just measure the mass and charge of everything that are present inside. So we can now see the challenge, like how can we tell that this is a peptide, and what the amino acid sequence for that peptide, by just seeing numbers from the machine. So first, let's go over how the mass and charge can be measured. So let me take you back to like high school physics maybe. If we want to measure the property of ion with charge, if it has charge, then we can accelerate the ion with charge with an electric field. If you apply a voltage, ions with more charge will run faster due to the electric field. So what you can do is you can accelerate all the peptides and protein ion, everything in your sample, with a certain voltage, and then you measure how long it takes for those molecules to travel through your mass spectrometer until it hits a detector at the other end. The amount of time it takes, you can convert back the amount of time, you can convert back to velocity, and from velocity you can do a conservation of energy from kinetic energy with mass time velocity square has to be equal to the electric potential that are applied to the ion. And the electric potential is a product between charge, like how many charge there are, and the voltage. So by solving this equation, there are only two unknowns. We know the voltage, that's what we apply to the machine. We know the velocity because we convert back from time. But we don't know the mass and we don't know the charge. So from the physics equation, the only thing we can deduce would be the ratio between mass overcharge. And that will be in the form of the voltage and the velocity squared. So this type of mass spectrometry is called a time of flight mass spectrometry. Because as the name implies, you are measuring the time of flight of ions from one end to the other end. And you use the time to calculate the velocity and then to deduce the mass overcharge. In some other mass spectrometer called orbit-trap analysis. So orbit-trap here is mean orbit. Orbit is when you have some molecules or some object like orbit over the other, like it travels in circle over the other objects. So in this case, we are going to accelerate the peptides of protein ion with magnetic field. And it's going to travel inside the mass spectrometer in orbital motion, in circular motion. And what we are going to measure will be the frequency of the orbit. If the frequency is high, like that means the molecule is moving very fast, like the frequency is high. If the frequency is low, then that means the molecule is moving slowly. We know the velocity again. So this orbit trap will convert information we have from the orbiting frequency of the ions back to the mass overcharge ratio. So based on physics, we can only measure this ratio. But to interpret which peptide it is, which amino acid it is, we want to know the mass, not the mass overcharge. We want to know the exact mass. So how do we tell if a peptide is plus 2 charge or plus 3 charge or plus 4 charge? We use chemistry. So mass spectrometry is a very fun technology. You have to use everything you learn from science. So from physics, we can measure mass to charge ratio. But from chemistry, because we know that proteins, peptides are organic. They have carbon, nitrogen, oxygen, sulfur as a component. And in nature, these atoms exist as isotopes. You have carbon-12 as the monoisotopic mass, but you also have some carbon, big carbon-13 with 1 extra neutron, and also carbon-14 with 2 extra neutrons, or nitrogen-15 with 1 extra with extra neutrons. So protein and peptide, they also exist as isotopes. And the mass spectrometer that we are using nowadays are so sensitive that when we measure the mass overcharge of a peptide or protein, we are not just seeing the monoisotopic mass, we are seeing all the isotopes. So if you zoom in the data that I show here, you will see that for each ion, you are going to see what they call an isotopic envelope. So the series of monoisotopic and then 1 extra neutron, 2 extra neutrons, 3 extra neutrons, all the way 4, 5, 6. And we can deduce from the difference in mass overcharge between 2 adjacent isotopes. Because we are measuring mass overcharge and adjacent isotopes differ by 1 neutron, so we know that the difference in mass overcharge between 2 adjacent isotopes has to be 1 overcharge. If according to this formula, if the mass is m and m plus 1, neutron is 1 unit, but both of them are divided by charge. So the difference that we are seeing the mass on the mass spectrometer is 1 overcharge. So in this case, in this example, it's 689.85 being the first mass. The second one is 690.35. This differ by 0.5. So this means the charge is 2 because 1 over 2 is 0.5. So we can deduce the charge from the isotope properties, from the isotope patterns. So combining these properties of organic compound with the physics of the movement of ions in electric or magnetic field, together we can solve for the mass. So this is the underlying principle behind mass spectrometry, how we can solve for mass for any compounds or molecules that we put inside. Okay. So one keyword that you are going to see when you study about mass spectrometry is how we transform proteins into gas. Usually, if you want to transform anything from an aqueous phase into gas, you have to introduce energy. The problem about biomolecules, protein, is that if you introduce energy to it, if you introduce too much energy, it's going to break apart. Biomolecules are fragile. So before we have mass spectrometry for proteins, people in the past, about 20, 30 years ago, they came up with two techniques to ionize or to turn protein into gas without destroying it. And there are two main ways to do this. One is called the electrospray, ESI, and the other is called matrix acid laser desorption, used like a matrix solution to absorb the energy, so that the amount of energy that got transferred to the protein is small enough to turn the protein into gas, but not so high that it destroys the protein entirely. So these two techniques are what we use to transform protein into gas. And these two techniques got the Nobel Prize from 20 years ago because of this capability. Okay, so let's say we want to study proteins. We can start from designing our experiment. Do we want to study every protein in the sample, like whole cell proteomes, or we can do also subcellular proteome? The nice thing is about protein. So when we study transcriptomics, when we isolate RNA from cytoplasm, we got everything because all RNA can be found in cytoplasm. But for protein, because some protein might be in the cell membrane, some might be in the mitochondria, some might work in the nucleus, some might work in a different organelle. People have a choice. You have a choice to study every protein. You want to extract everything, or you want to enrich like certain organelle or certain part of the cells, and study only those proteins. So some people, if they are interested in cell membrane, they can focus on the protein that are embedded into the membrane, and they can elute out all the cytoplasmic proteins. So you have a choice when you study protein, which groups of protein you want to focus in. You can even isolate certain protein complex. Let's say you want to focus on certain protein specifically. You can take the sample from the cytoplasm, you can run the gel, and if you know the size of the protein, you can even select just that part of the gel that corresponds to your protein and do the mass spectrometry analysis. So you have a choice to do any specific or targeted analysis based on your knowledge. When we want to study protein, you will see later on that if we analyze protein, intact protein as a whole, because proteins are quite large, the spectrum that we are going to get will be complex. So usually if you want to identify the protein, we can do concepts similar to DNA sequencing. We can cut the protein, like how we fragment the DNA into the library for sequencing. We can digest the protein with protease enzymes to generate smaller peptide, which can be easier to analyze in the mass spectrometry. And then we can do the bioinformatic analysis to identify which protein are found by looking at the sequence from the peptide that we detected. So there are many choices for protease. Each enzyme has different specificity. So usually people use trypsin because this is the enzyme that our body used to digest protein already, so it's very potent. But trypsin has a specificity that it only digests after arginine or lysine. So if your protein doesn't have arginine or lysine, some protein might not have those amino acids, trypsin is not going to be effective. It's not going to cut anything. So some people suggest that you should look at the protein first and see if your protein has the cleavage site for the enzyme. And you can pick the enzyme accordingly. And some people even suggest that we can use multiple enzymes. Why don't we just use multiple enzymes to generate a more diverse set of peptides and then analyze the data. Then we get better coverage of the protein. Because some peptide might not cut, some protease might not cut well in certain region of the protein. So this kind of depends on how much coverage you want to detect the protein. Okay. So when I mentioned that the study of full kind of intact protein can be difficult because it's very large, but there are also benefits in studying full intact protein versus if you digest it. So people call this either top-down, which is to analyze the intact protein, or bottom-up, when you digest the protein and analyze the peptide. So for bottom-up, the benefit is that when you have smaller peptide, you have a simpler mass spectrum that you have to analyze. So we can identify the peptide more easily, more confidently. But the limitation is, let's say you want to study the function of a protein, and this protein can exist in multiple isoforms, depending on the PTM, depending on the post-translational modification. Let's say the protein has to be maybe phosphorylated at 2 locations for it to be functional. If we digest the protein, we might detect phosphorylation on a peptide, but we have no idea if on the same protein is the other side also phosphorylated. When we do bottom-up, when we digest, when we cut the protein into small pieces, we no longer have the entire picture of post-translational modification. So if your analysis hinge on the fact that you can determine the isoform of the protein, you have to study the intact protein, because with the intact protein, you can see 2 phosphorylation on the same molecules, because they will appear in the same mass spectrum. So that's the only way that you can distinguish protein species from the other. But to analyze protein as an intact molecules, if it's larger than, let's say, 50 to 100 kilodalton, then you probably cannot interpret the mass spectrum, because it will be too complex. So there's some limitation there. So it's a trade-off between the difficulty in the analysis versus the ability to distinguish protein species. Okay. So these are the choices that you have to make even before you design your proteomics experiment. You have to think about this factor. It's a good thing, because you have more choices than if you perform transcriptomics. You have more choices about which protein to focus on. Do you want to focus on identifying the protein, or confirming the PTM, or distinguishing protein species? Okay. Another factor that makes proteomics difficult, there's always more factors that make proteomics difficult. And this is because mass spectrometry, the process of measuring the mass overcharge, is a physical process. You have to accelerate the ions to move and measure the time, measure the frequency. So it takes time. It takes time to scan the molecules. So imagine if you dump all your protein and peptide from the whole cell at once into the mass spectrometer, it's going to be able to scan maybe 10% of the proteins, and then all the protein will degrade. And you will lose the rest of the information, because the mass spectrometry cannot scan the protein fast enough. So the trick is we have to gradually introduce the protein and peptide into the mass spectrometer at a time. And one technique for doing that is the chromatography. We know that chromatography is used to isolate a mixture of compounds based on certain properties, so that you can get each compound one at a time. So this is the design of a mass spectrometry experiment. You isolate your protein and peptide. You have to run those samples through the chromatography, usually liquid chromatography. Then peptide and protein will flow into the mass spectrometer, a small number at a time, so that the machine has enough time to analyze everything. Another technique that you can use to help the machine, help the mass spectrometer, you can fractionate your sample. You can differentiate your protein and peptide, maybe based on pH, maybe based on the charge, so that each of your aliquot is going to have different set of protein and peptide, and you can feed kind of one fraction to the mass spectrometer one at a time. So fractionation and liquid chromatography together help reduce the complexity of the proteome, so that the machine has enough time to analyze everything for you. Sometimes that's not enough. Sometimes you want to study a certain post-translational modification, and in general, PTM are quite rare. If you have a certain number of molecules of the protein, only a small fraction will be modified. So if you want to analyze PTM specifically, just diluting your sample into multiple fraction alone might not be enough. You have to enrich for certain PTM. And because different PTM has a different chemical property, people have developed different assays, different columns, different techniques to enrich different type of PTM. So this is also something that you have to decide before running the mass spectrometer to isolate or to enrich certain PTM beforehand. Okay. So based on these techniques that we need to simplify the protein sample so that the machine has a chance to scan or analyze everything. This also makes proteomics very time consuming. So let's say we have 3 patients and 3 controls. This is a very small scale study. To analyze every protein in human, we probably need to divide the sample into 10 fractions. Actually more, but 10 is like the minimum. And to analyze protein and peptide within each of these 10 fractions, you have to run the chromatography for 2 to 4 hours to separate out all the protein and peptide. So how much time do we need for this cohort? You need 10 days just to analyze all fractions for all the samples. And this doesn't even count like replicates or if you have a larger cohort. So compared to DNA sequencing, this is 10 or 100 times slower. But this trade-off, like if you want to analyze protein, this is the base technology that we have. Okay. So up to this point, like we saw the process where protein and peptide that we isolated are passed through the chromatography and then goes into the mass spectrometry. So now we're going to look through like, the journey of the protein and the peptide as it went inside the machine. Like, what information will you get back from the machine? So the first step, the machine is going to measure the mass overcharge. That's what the mass spectrometry is for. It's going to measure the mass overcharge of everything that it can measure as these ions, protein and peptide, went through the machine. So the x-axis here is the time that the protein and peptide came out from the chromatography. So these are early peptide, early protein. They pass through the chromatography very quickly. And at the end here, these are the protein and peptide that can move slowly through the chromatography. So this might be like large protein, large peptide, might be hydrophobic, might be depending on how you design your chromatography. Peptide with certain property will flow through very fast, and peptide and protein with certain properties, certain chemical properties, will flow through very slowly. So we can isolate different protein and peptide through chromatography. So the first thing that we can see from the mass spectrometer is this map. This is a map of mass overcharge of everything that the model can see, that the machine can see, as the sample passes through the chromatography. And then, at each time point, if we look at the cross section, this is the spectrum that I just showed earlier. So this is the profile at a certain time from the chromatography. These are the protein and peptide ions and also contaminant and everything that came out at that time. So some of them might be highly abundant, some of them might be very low abundant. So one of the assumptions that people make, because we have no idea if these are peptide proteins or contaminant or anything, one assumption that people make is that the proteins, because we are analyzing protein, the exact protein from the sample, we expect protein and peptide to be the most abundant in the data. We don't expect contaminant to be more abundant than the protein and peptides. So what happened inside the mass spectrometer, and this is called a data dependent mode. So the mass spectrometer will see the profiles of masses, and then it will isolate, it will select certain mass overcharge that are abundant from top to bottom. And for each of them, the machine is going to isolate these ions and then analyze them further. We will talk about how this will be analyzed further. So with a data dependent mode, the machine will rely on the pattern of mass overcharge that came out and assume that the abundant ones are interesting and then select these for analysis. But this creates a problem. If the proteins that are important for the biological process or related to the disease are lowly expressed, not highly expressed, the machine will completely ignore them because it will assume that the abundant ones are more interesting. So people came up with a different technique called a data-independent analysis. So in this data-independent mode, the mass spectrometer will isolate everything, not just the most abundant signal. The data-independent mode, the machine will only isolate the most abundant signals, and this might cover the protein of interest. So in the data-independent mode, the machine will try to analyze everything, but we know that the analysis in the mass spectrometer takes time. So if you want the machine to analyze every individual ions, there's not enough time for that. So in the data-independent mode, we have to make a choice. We have to make a sacrifice. The machine, instead of isolating individual protein and peptide ion, is going to isolate a window of mass overcharge. So this blue area, red area, green area, brown area, and purple area. So within each area, they're going to analyze all the ions, all the protein and peptide inside that area. So you can imagine that the result, with the data-independent mode, each spectrum that we get as a result will correspond to one species of protein and peptide. But for data-independent mode, each spectrum that we get as a result will correspond to a collection of protein and peptide. So the data will be slightly more complex, but it allows the machine to collect everything. So the problem now comes to the bioinformatics part. How do we decode spectrum that have a mixture of maybe 3, 4, 5 protein or peptide together and identify all the individual protein and peptide inside? Okay. But before we worry about that, let's look at the process, how the machine isolates. When the machine isolates a certain protein and peptide, what does it do to generate the fragment that we will use to deduce the amino acid? So this process is called tandem mass spectrometry, because we're going to perform mass spectrometry analysis twice. The first time to measure the abundant protein and peptide, and then we isolate that. And the next mass spectrometry analysis will be to analyze the fragment that will generate from those individual protein and peptide. So the idea, how we generate the fragment from a protein and peptide, we view the protein and peptide as a polymer, with peptide bond as the chemical bond that connects individual amino acid. So if we can introduce the right amount of energy that can break the peptide bond, but not other bonds. The other bonds have higher energy. So if we can introduce just the right amount of energy, we can create the fragment of the proteins that occur only at the peptide bond. And that will create some consistency. If you can break the protein at any molecules, you have so many possibilities. But if you break just the peptide bond, then you can only generate so many different fragments. And the way that we do that in the mass spectrometer is we accelerate the protein and peptide and make it run into inert gas, nitrogen gas or helium. And when there is a collision between protein and peptide with the inert gas, the energy from the collision will transfer into the chemical bond, and it can break the peptide bond. So this is how we induce the fragmentation of protein and peptide. So just to show you how the product from the fragmentation correlate to the spectrum. So let's say we have this peptide, FQSADET. Let's say we accelerate this peptide. There are many molecules for this peptide, and they will run into nitrogen gases. The first molecule of the peptide might break at this peptide bond between Q and S. So the product will be FQ as one molecule, and SA all the way to the end as a second molecule. Maybe a different molecules of the peptide breaks here between S and A. So the product will be the first 3 amino acids as one molecule, and the second part as a second molecule. And the masses overcharge of all these possibilities are what being captured in the mass spectrum. So some of these ions might correspond to the F amino acid alone. Some might correspond to A and R, which is the last two nucleotide amino acid. Some might correspond to F and Q. Some might correspond to FQ and S. Some might correspond to FQSA. So this spectrum that we get after fragmentation has all these signatures that we need to deduce, that we need to decode to identify F, and then Q, and then S, and then A to get back the entire amino acid sequence. So this is the process of peptide sequencing. And in theory, it looks like this. We have a spectrum, and we don't know the answer, which peptide it is. But we have to look at the mass overcharge, and what we want to see are these gaps between two ions. If these two ions are adjacent fragments, the mass difference between them should correspond to the amino acid, as in the logic flow that we talked earlier. If the weight of Q is 128.06, we would see two fragments, one with F and one with F and Q. These two fragments will be separated by exactly 128.06 apart. And we can deduce that there must be Q in amino acid Q in the sequence. And if we can continue this pattern, we see a gap of 87.03, a gap of 71.04, we can deduce the next amino acid to be S, and then the next to be A, and the next to be T. We can deduce from this pattern. But this looks easy, if I tell you the answer, that these are the correct ion to interpret. But who knows, when we get the spectrum, who knows to skip this one, the most abundant one? Who knows to skip this, who knows to skip this, who knows to skip this. So the difficulty comes from the fact that we don't know which ions to interpret when we have the mass spectrometry data. But still, if you're curious, if you really go into a proteomics courses, you will learn how to do this by hand. So this example that comes from the homework when I was a PhD student. We have this spectrum, and we have to deduce that this is the peptide VAT, and T has the phosphorylation, and then PVDWK, just based on the numbers that we see on the screen. It's possible because there's not many ways that you can create peptide that can produce this kind of pattern. But you cannot do this for 10,000 spectra. Each mass spectrometry run will generate more than 10,000 spectra for you. So even though human can do this, given enough time, it's not feasible for high throughput data. So what we do instead is a database search idea. So we have a new spectrum where we don't know what the answer is, but we have two possibilities. If we know that this peptide or proteins, let's say, come from human or come from mouse, because we have a good understanding of DNA, RNA, and protein in human and mouse, we can generate the possible, the universe of possible amino acid sequence that we would see if we study human or mouse. And if we can generate all these possibilities, all these possible amino acid sequence, because we know when we fragment the peptides, we generate the ion like IE, IEL, IELL, right? So we know what will be the mass overcharge that will be produced. So given the known sequence of amino acid, we can generate what would be the possible spectrum, what the mass overcharge should be. And then we can compare this expectation to the data. And if the mass overcharge can all line up, then that might be our answer. But you have to go through this for all the possible protein and peptide. Each one you generate the expected spectrum, and then you compute the similarity between the two. It's a very brute force process, but it gets the job done. You can interpret your spectrum by asking which possible protein and peptide produce the theoretical spectrum, the expected spectrum that match my observation. Another possibility if you study model organisms, other people might have analyzed it before. So you have previous proteomics data with annotation. People know which spectrum corresponds to which peptide. So we can also compare spectrum to spectrum. If the new spectrum looks like a known spectrum, then we can transfer the amino acid sequence from the old one to the new one. So we have two different kind of database search. One search against the possibility of protein sequence, amino acid sequence. The other one search against previously known spectrum. Both of them have the same limitation. You only get to identify known protein and peptide. So if you're analyzing new species, if you are analyzing cancer mutation, there's no chance that you can find it in the database because you're not going to see new mutation. And you might think that when we analyze DNA sequencing, we perform BLAST to map, and we know that BLAST allows for mismatch. Why can't we do the same thing for mass spectrometry? The problem is when you digest the protein into peptide, usually the length of the peptides are 5 to 20. So you only have 5 amino acids up to 20 amino acids. If you perform BLAST and you allow mismatches, you're going to get so many possibilities because you only have 5 to 20 characters. If you allow 1 or 2 matches, it can match to almost everything in the database. So we cannot do things like BLAST for proteomics because of that. Another headache is the search for post-translational modification. Because for each protein and peptide, not all of them are modified. So let's say you have this peptide in your sample, and you want to consider if this peptide can be modified, and you allow for oxidation of methionine and phosphorylation of serine and threonine. Because each peptide might be oxidized but not phosphorylated, or phosphorylated but not oxidized, or phosphorylated at both sides, or phosphorylated at one of the sides. So starting from one peptide, your database already have to have 8 possibilities to account for all possible different modifications, which gives you different masses on the peptide, which gives you different spectrum. So searching for PTM expand the set of possibilities by more than tenfold. So searching for PTM is also not easy. So nowadays, similar to how nanopore technology use machine learning and AI to interpret the signal, we also try to understand mass spectrometry data by using machine learning and AI. So this is called de novo peptide sequencing. We are not going to use database. We're going to make the AI look at the raw mass spectrum and try to identify. For example, there is a sequence of LD and V in the middle of the spectrum. So we know that whatever the sequence, the answer would be, it have to have LDV or VDL in the answer. So this can allow us to remove possibility. We don't have to consider all possible proteins. We just need to consider the one with these signatures inside the sequence. But the goal for full de novo sequencing is to have the AI identify the whole sequence from scratch. So nowadays, this is still not reliable. There are several approaches, and I think we are getting there, but maybe now it's not good enough. So when people analyze proteomics data nowadays, we still fall back to database search approach. Okay, so once we identify peptides, we want to map them to proteins, which we already know how to, and then we want to quantify the protein. It's not so different from transcriptomics. In transcriptomics, we cut the RNA into fragments, and when we get all the reads, we map back to the RNA, and then we count number of reads. For peptide protein, it's the same principle. We cut protein into peptide, we measure the abundance of the peptide, So when we map the peptide sequence back to the protein, we just add up all the intensity, all the number of ions, the number of molecules for the peptide. But there are some differences that I want to highlight. First, there is no concept of read count for protein and peptide. But instead, some people count the number of spectra, because if we have abundant protein or peptide, the mass spectrometer will encounter that protein and peptide several times. So it will generate more than one spectrum for that protein or peptide. So the number of spectrum is also a proxy for the abundance for the protein and peptide. The intensity, which is an approximate of the ion count or the molecule count for the peptide, can also be used to measure the abundance of the protein. But if we think back to transcriptomics, when we measure the read count, one thing that we worry about is the PCR bias. Some sequence can be amplified better than the other, and longer gene will produce more read by chance. For protein, the problem is even more complicated. When we turn the protein into gas, when we turn the protein peptide into gas, the ionization process also have a bias. Certain peptide and certain protein will turn into gas very easily, but some protein and peptide will not. So this is similar to PCR amplification, except we kind of can estimate the bias in PCR amplification, but we still have no idea how to estimate the bias based on ionization. So there's no correction for ionization efficiency in mass spectrometry data, because we don't yet know what factor to account for. So when people quantify the protein, this weird thing came up. When we analyze transcriptomic data, we count all reads that map to the gene. But in proteomics, most people will not count all peptides that map to the protein. So when you read the literature, you might be confused, why? And part of it is because there are many biases, like ionization bias, that cannot be corrected. So some people found that empirically, by trial and error, counting only the top most abundant peptides, maybe top 3, top 5, actually gives you a better quantification of the protein. So this is based on trial and error. They found that doing this is better than counting everything. Okay. But regardless, if they count all the peptides or some peptides, what we know from the nature of the data, the intensity or the ion count for the peptide and protein, these numbers are also log normally distributed. So it behaves like nanostring and microarray data. When you take the logarithm of the intensity, you can apply statistical model that assume normal distribution, like t-test. So the data distribution is easy to model, unlike read count. Okay. Protein also have isoform issue, like DNA and like RNA. If you have protein that share the same exon, and you identify peptide that map to that exon, you cannot tell if it's from isoform A or isoform B. In proteomics, people come up with different ways to handle this. So because in proteomics, you cannot normalize precisely every effect. So many of the analysis techniques became empirical. People try several ways, and then which one that works well became the standard of the field. So for example, some people suggest that we can assign the peptides that are shared between isoforms to the one with the highest abundance. It's like a winner takes all. So you assign everything to the most abundant isoform and let it win, which is weird. The second idea is to distribute the shared peptide proportionally. So we assume that if we have peptides that are unique to each isoform, those give you the proportion of the abundance between the isoforms. Can we distribute the shared peptide according to that proportion? The problem is sometimes you don't know the proportion because many proteins will have so many shared exons that it's difficult for you to detect the unique peptide. So idea 2 doesn't work kind of all the time. So right now, people are starting to navigate, to gravitate, toward the idea number 3, which is to disregard, like to remove all shared peptide and only trust in unique peptide. So all the shared peptide will not be count, only unique one. So people are moving toward this idea of if we cannot solve this problem, just not make it more complicated, just disregard the data. And one thing that you will see for proteomics, instead of reporting abundance per protein, they're going to report abundance per protein groups, okay? A group of multiple proteins. So in this case, when you see protein groups, it means that these are proteins that cannot be distinguished from each other based on the peptide that you can detect. So these proteins share the same exon, and based on the peptide that we can see, there's no way to tell them apart. But still, we don't want to throw out all the signal, all the data. So we will report this protein as a group, and we consider the shared peptide as the intensity for the whole group. But we don't know which protein exactly that are expressed. So similar to transcriptomics, in transcriptomics we have RNA sequencing, which sequence every RNA. And then we have nanostring and microarray that target certain genes, certain isoform. For proteomics, it's similar. If you do mass spectrometry as a discovery analysis, you want to identify every protein and peptide. But once you got your biomarker, you know that these are the proteins related to the disease, I want to measure it in my future patient or in my future sample. We want accurate quantification of the protein, of the peptide. So in the discovery analysis, in mass spectrometry, you measure the abundance of the peptide, of the whole peptide, only once. But if you want to get an accurate measurement of the abundance of the peptide, the better thing you can do is once you fragment the peptide, you generate all the fragment of shorter ion from the peptide, you also have the abundance for this ion. So this peptide, if you fragment it between E and P, between P and T, between T and I, you generate these 3 ions. You have the abundance for these 3 ions as well. So the better measurement would be for you to measure the abundance of this peptide as a function of abundance for these ions. So you kind of make multiple measurements, one for each fragment ions, and together they give you a more accurate estimate, overall estimate of the peptide itself. So people call this multiple reaction monitoring techniques. And then as the name implies, instead of measuring the peptide only once, you're going to monitor the fragment ions that are generated from this peptide. So there are multiple reactions. You fragment the peptide, the blue peptide is your target. You fragment the blue peptide, and then you isolate one of the ions, one of the fragment ions, to measure the abundance. You fragment the peptide again, you measure another ions, and then quantify it. So you get 3 measurements from 3 reactions. Each will tell you, these together will give you a better estimate of the abundance of the blue peptide. So they use different type of mass spectrometer is good for different analysis. Certain type will be good for discovery, like the one that we talked about earlier. But some of them will be good for multiple reaction monitoring. So in mass spectrometry laboratory, if they want to do both discovery and targeted quantification, they have to have 2 different types of mass spectrometer inside. Okay. So far, we talked about mass spectrometry on one sample. But we know that when we analyze data, we want to compare effect across samples, like across treatment conditions, for example. We have to be able to compare protein abundance across samples. And the problem with mass spectrometry is the chromatography is very noisy. If you run 2 chromatography, it's going to look different, even if you put in the entirely the same sample. You're going to get different chromatography because it's just the nature of the flow of the process of separating all the protein and peptide. So in the past, with no better option, we have to compare the liquid chromatography profiles between experiments, and we hope that the same protein and peptide with the same mass overcharge will come out at the same time or similar time. So we might allow plus or minus 5 minutes, and hopefully we can see the same protein peptide coming out across experiment, across runs. But this is not reliable. People found earlier on that this is not reliable. If you want to repeat your previous analysis, or if your lab buy a new liquid chromatography column, you can no longer compare from experiment to experiment. So you have some kind of a labeling technique. If your sample is a cell culture, so you grow the cell over multiple generation, you can feed it with food with heavy isotopes. So in one culture, maybe your control condition, you feed it with no isotopes, just a monoisotopic version of the food. But in your treatment, you might treat it with arginine 6, so it will be a heavy arginine with 6 extra neutrons. So what happens is your cells in the second culture, in your treatment condition, is going to incorporate heavy arginine into all the protein that it produces. So after a long period of culture, when you extract the protein and peptide, all the protein and peptide that have arginine in the treatment condition, all the protein and peptide with arginine will be heavier than the same protein and peptide in the control. So this allows you to mix the protein and peptide from the two samples together and run only one mass spectrometry analysis. And what the bioinformatics software has to do is to identify two pairs of mass overcharge that have similar spectrum and have the expected shift in the mass. If this peptide has one arginine, we expect the gap to be 6 Dalton. If this peptide has 2 arginine, we expect this gap to be 12 Dalton. So the bioinformatics software not only has to deduce the peptide sequence, it also has to check whether the deduced sequence match the difference in mass between these two peaks. If the number of arginine doesn't add up with the difference in masses here, then that means something is wrong. So the bioinformatics tools has to be more sophisticated to handle this. But this allows you to remove all the technical bias because you can analyze your sample together and get a single mass spectrometry output. Okay. But this only applies to cell line. And you might ask, why do we use arginine? This is because people use trypsin to digest the protein. And trypsin will cut after arginine or lysine. So by feeding cells with heavy arginine, peptides that are cut with trypsin, half of them will end up with arginine, half of them will end up with lysine. So people usually use arginine or lysine as the isotope because when we use trypsin to cut, we are guaranteed to have these amino acids inside. But this only applies to cell line. You have to feed it long enough until it incorporates the isotopes. But if you are analyzing blood sample, you cannot culture. You have to analyze it as it is. So the other technique that people came up with is called tandem mass tag. And this is very clever. So what we do is for each sample, let's say different blood sample, we extract the protein, digest the peptide, with the digest the protein, we do whatever we need to do. And then these tandem mass tag molecules, these are chemical molecules that can be attached to the N-terminus of peptide. Any peptide is attached to the N-terminus. And these TMT tags comes in different variety. So each variety will have 2 components. So the entire molecules, the entire tag is one molecule, but in the middle of the molecules, it has a bond, a chemical bond that can be cleaved or has low energy like peptide bond. And the design is different species, different like variety of the TMT tag, the location of this bond that can be cleaved will be different. So when this bond is cleaved, it's going to produce a product fragment that have different mass. So for example, in this example, we have a TMT with 126. So this means that the reporter ion here is 126 Dalton. TMT 127 means that this reporter part is 127 Dalton. But the entire molecules weigh the same. So what happened with this design is when you tag a peptide from sample 1 with certain type of TMT, peptide from sample 2 with a different type of TMT, and peptide from sample 3 with a yet different type of TMT, and you mix this sample together. In the first mass spectrum, when we see the intact protein or peptide, the same peptide from all the sample are going to have the same mass, which will be the mass of the peptide plus the mass of the intact TMT. So they will line up at the same mass overcharge. So when the mass spectrometer isolate these ions, all peptides, these same peptides coming from different samples, will be isolated together. They have the same mass, so they will be isolated together. But inside, doing the fragmentation, so some fragmentation will cut the peptide bond inside the peptide or the protein, but some fragmentation, the collision, can induce the cleavage of this reporter. So what happens inside the fragmentation in the spectrum? If you zoom in on the 125 Dalton to 130 Dalton, you're going to see these peaks each correspond to different reporters. One for one, the smallest for 126, 127, 128, 129, 130, 131. So essentially, we have the relative abundance of the peptide which come from different samples in the same spectrum. So this also reduces all the technical bias. You analyze peptide from all the patients together, and you get these profile to show that this peptide for patient 1 is very low. Patient 2 and patient 6 are the same. Like patient 3, 4, 5 are high. So you can tell the expression level, relative expression level from the mass spectrum directly. So nowadays we have tandem mass tag, I think with up to 12 or 20 species. So you can multiplex your mass spectrometry in a single one. Okay, so up to this point, I cover the majority, the main part of proteomics with mass spectrometry that focus on protein expression. So for the last part, I would like to introduce a clever way, a creative way to analyze protein structure using mass spectrometry. So the first technique is the simplest one. So let's say you want to understand the structure for this protein complex. You want to understand which proteins are bound together in this complex, which are next to each other. So one thing that we can do with chemistry is we can introduce a cross-linking compound. So this cross-linking can recognize two different amino acid residues in the structure. And if the two residues are near each other in 3 dimensions, this chemical cross-linking compound can induce a covalent bond inside the molecules. So it will create like a covalent connection inside your complex. So once you digest your complex, some peptide will have this cross-linking in between. So it will be like 2 peptides connect to each other. And when we run mass spectrometry, we can identify which peptides are cross-linked together based on the mass spectrum pattern. So this will also require a specialized bioinformatics algorithm to identify not linear peptide, but 2 peptides connected to each other. But it's possible. So once you identify which peptides are connected to each other, if you can map like one peptide to the brown protein, one peptide to the purple protein, the fact that they can be cross-linked means that the brown protein and the purple protein has to be next to each other in 3D. So we can use this information, like which part of protein A is next to which part on protein B, we can use this information to drive the structure simulation. When we model the 3D structure, we can use this information as a concept to say that this protein and this protein, they have to meet each other at certain location. So that can help the modeling process. The second technique is called hydrogen deuterium exchange, and it utilizes the fact that deuterium, which is the heavy form of hydrogen, is very reactive. If you introduce like a water but with deuterium instead of hydrogen, any hydrogen in your protein that are solvent accessible, the water can reach, it will change into deuterium. So let's say you have a protein, and you know that when it's phosphorylated, there will be a change in structure. Maybe the enzymatic domain will open up to find the substrate. And you want to understand what's the part of the protein that open up to recognize the substrate. So what you can do is you can do a deuterium exchange. So you introduce the water with deuterium into the protein sample. And what you will find is that if the protein, certain part is closed and it's not accessible by deuterium, the peptide will be in, let me try to find my cursor a little bit. Okay, looks like I lost my cursor. Okay, let me keep explaining. So the part of the protein that is not accessible in the, let's say in the bottom protein, some part is closed. So the middle hydrogen will remain hydrogen, but the external peptide and protein, all hydrogen will be substituted with deuterium. Compared to the top isoform, when it's phosphorylated, the protein opens up. So every part is now accessible, and every hydrogen will be substituted with a deuterium. So when you compare mass spectrum from the top condition and the bottom condition, like phosphorylated and non-phosphorylated, you will be able to identify peptide that remain hydrogen but became deuterium across the two conditions. And whichever part of the protein that those peptides can be mapped to, like the peptide that I highlight in yellow, whichever part of the protein that this peptide can be mapped to, those are the parts that opens up when this is phosphorylated. So we can understand the change in protein conformation, the change in solvent accessibility by probing how deuterium replaces hydrogen inside the protein. So this is one of our techniques. Another technique that's also interesting is based on the fact that when we measure the mass overcharge of the protein, like using the time of flight technique, like we let the protein and peptide flow in vacuum through the electric field or magnetic field. Another factor that can affect the flow is the structure of the protein. If our protein can form dimer, trimer, tetramer, it will get larger and larger. The size of the protein can change the amount of time that it takes to flow to the mass spectrometer. So this time of flight technique can also be used to distinguish change in protein conformation for the intact protein or the polymerization of the protein by seeing the time it takes for the protein to flow through the machine. Okay. So just to give you an idea that we can also adapt mass spectrometer to study different aspects of the protein, not just the expression level. And also, it's not a scope of this class, but you just want to know that the concept of mass spectrometer, like breaking down the ion, identify the signature, and then use database search or algorithms to deduce the compound that are inside the sample, is not limited to protein or peptide. It can be applied to any molecules, such as lipids or metabolites. So if you study metabolomics, they're also going to use mass spectrometry. The liquid chromatography part will be changed to gas chromatography, just to accommodate these molecules. The fact that proteomics are maybe more popular is because protein and peptide, they are polymers. So their product, the fragmentation product, are easier to understand. But if you have a chemical compound, like if you have a lipid, it's more difficult to tell like which part can break by the energy and what would the signature be. But what you can do always is if you have a library of known compounds, you can run the mass spectrometry on known compounds and you have the spectrum. So when you have a new spectrum from your sample, you can compare to the library of known compounds. And if the spectrum looks similar, then you deduce that maybe that's the compound that are in the sample. Okay. But still, like the same limitation with proteomics, the interpretation of the spectrum can be confounded by noises or contaminants from multiple compounds in the same spectrum. Okay. So that's kind of everything I want to talk about today.